Good evening. Welcome. My name is Laura Heisler, and I am the Director of Programming for the Wisconsin Alumni Research Foundation and the Director of Outreach for the Morgridge Institute for Research, which are two of the three organizations that collaborate to bring you this Crossroads of Ideas series, the third being the Wisconsin Institute for Discovery. And we are delighted to be able to bring you this series, which focuses on subjects that matter to all of us in our lives that also happen to be the subject of groundbreaking research here at UW-Madison. This is the second installment for this semester, and we are pleased this semester to be able to celebrate a remarkable anniversary. It was 20 years ago that Jamie Thompson, who is a researcher here at the Morgridge Institute for Research upstairs, as well as UW-Madison, had his groundbreaking discovery regarding the propagation of human embryonic stem cells, which forever changed science and the world that we live in. And what we're doing this fall is taking a two-part look at the implications for us, tonight focusing on the ethical landscape, and in the next month on November 13th, focusing on where the field is likely to go and looking out ahead to what we have in store over the next 20 years, thanks to the groundbreaking discoveries that started right here at UW-Madison. We're going to be, I want to give you a couple of housekeeping notes for how this session is going to go. We will hear from each of our three speakers very briefly, and then they'll have a conversation amongst themselves, teasing out some of their different perspectives on the topic of where things stand with ethics and stem cell research. And then there'll be an opportunity for your questions. And so when we get to that point for your questions, we do ask that you raise your hand and wait for one of our mic runners to reach you, because we record these sessions. You can find the recording on the discovery.wis.edu site in a few days. But that means that it's really valuable for us to be able to hear your questions so that it makes it onto the recording. So please do wait for that. And so then you will have a chance to have some conversation with the presenters, and then we're going to end promptly at 8. And if you still have some questions, they've all agreed to stick around for a few minutes if you want to follow up on something that you didn't quite get a chance to ask. So I would like right now to introduce our speakers with no further ado. Our first speaker will be Anita Baracharya, who is an assistant professor of cell and regenerative biology at the Weissman Institute here at UW-Madison. She's also going to be moderating the discussion. We also have Chris Saha, who's an assistant professor of biomedical engineering and an investigator here in the Wisconsin Institute for Discovery upstairs, the twin institute, one of the twin institutes, along with the Morgridge Institute for Research. And then we also have Dietram Scheufele, who is the John E. Ross Professor of Science Communication in the Department of Life Sciences Communications here at UW-Madison, as well as an affiliate at the Morgridge Institute for Research. So with that, I'm delighted to welcome Aninia Bhattacharya.
Thank you, everyone, for coming tonight. And first of all, I want to apologize. I'm getting over laryngitis, so I'm hoping that you can hear me. This isn't my normal voice, though. As Laura said, it was 20 years ago that Jamie Thompson first isolated human embryonic stem cells from human embryos. And it is important for us on this anniversary to pause and think about the ethical and social and political and scientific impact that this discovery had. And as Laura said, that will be the topic of today and then also next month's Crossroads series. First, I'd like to give some examples of the impact that this discovery had in the scientific community, and then talk a little bit about the ethical implications that initially were brought up. Excuse me, this is the front of Science magazine that puts human stem cells as the technology of the year, immediately after Jamie Thompson and Jeff Jones here at UW isolated the human embryonic stem cells from human embryos. Excuse me. And this discovery scientifically launched two fields, really. The first was human stem cell biology, and the second was regenerative medicine. And in this paper, Jamie Thompson and Jeff Jones isolated 5 human embryonic stem cells from human embryos, and these 5 lines are still being distributed worldwide today by Weissel. And this graph just shows that over the last 20 years, the distribution of these human embryonic stem cells has increased and has not decreased very much, even in the last 10 years. In addition, having these human embryonic stem cells in the scientific community it caused a huge new research field, studying human stem cells, studying what they could do, studying what made them unique, and studying them to find out how we can turn other cells, adult cells, into cells that look like human embryonic stem cells. And this graph shows the number of publications that have occurred in the last 20 years that are related to human stem cells. And also for isolation of these human embryonic stem cells, launched what we call regenerative medicine. That is the ability to take human stem cells and use them as a way to regenerate or replace damaged or diseased tissues. So in addition, at the time of this discovery, it was very new, obviously. Dolly the sheep had been cloned, but the fact that this was these cells were isolated from human embryos really took the public by surprise. It took many scientists by surprise. And so because of this novelty, there wasn't a good framework for how to do the science, how to talk about this kind of science, and really how to move forward. And so the discovery really captured the imagination of the public. There were many stories in the popular press about human embryonic stem cells. And as I said, there wasn't a good framework, especially at a regulatory level, for how to work with these stem cells. And because the discovery was done here, much of the initial infrastructure on how to move forward was done here by Norm Foss, Alto Charo, and their colleagues in trying to think about what is the best way and the most thoughtful way to move this science forward. And so over the last 20 years, there have been many guidelines that have come about from the National Academy and from international stem cell communities to provide a framework for scientists to move forward in their research, to do it in a thoughtful and a way that has some limits and also can potentially speak to some of the concerns that the public may have. However, these kinds of guidelines do not allay some of the concerns that the public has. We're talking about using human embryos, and so of course there are going to be ethical implications for that. And so today I think we'll talk a little bit about many of the social, political, and implications of this work. I will say that in addition to the public, more generally, the human stem cells also had an impact in the political arena. Both Presidents Bush and Presidents Obama had to make policy regarding the human stem cell discovery and how scientists, at least in this country, could move forward. And as you may know, their policies were slightly different, and with human stem cell research being the political arena, there is the prospect that it may continue to change in the future. So a lot of the initial concerns, the initial topics that arose from this initial discovery continue to be talked about today. But in addition to that, in the last 20 years, there have been new technological advances that build on or will work with the human embryonic stem cells. So, for example, the way we culture these cells in the lab, in the dish, has improved quite a bit. So now we can culture them in a way where we can start to put the cells together and make the cells look like human embryos again. So these are kind of synthetic human embryos. And so these raise concerns about what are we doing with these cells. There is also the more recent technological advance of gene editing with CRISPR-Cas9 that allows us to change the DNA of these human stem cells. And while scientifically this is really cool, we really need to think about what kind of impact being able to do this can have. And also, many of you have probably heard in the popular press about what we often call stem cell tourism, the use of the stem cells in different kinds of regenerative medicine, in the ability to provide therapy for different diseases. And while many scientists are thinking about these questions, many of the clinics that have arisen are not based necessarily on good science and can really take advantage of the public's need and want to have a disease fixed. And so that's also something we can talk about. And so with that, I would like to invite my colleagues to come up here, and we can talk a little bit more about the issues, apart from the scientific issues, that are ongoing right now.
Sure. Let me just expand really quickly just on a couple of things that you said. And Laura said early in the introduction, my work in Wisconsin focuses mostly on the political social dynamics surrounding emerging technologies, and stem cells certainly since coming back to Wisconsin as my PhD here a long time ago, went off to Cornell to become a professor and then eventually came back here. Stem cell, of course, is one of the big issues here. It falls actually into a broader set of scientific issues that some people at the National Science Foundation better. I can't get a lot closer without brushing up against it. A lot of people at the National Science Foundation call ENBIG technologies. So ENBIG stands for nano, bio, info, cogno. So this interface of emerging technologies at the nanoscale, molecular scale, merged with biology, merged with information science, neuroscience, hence the ENBIG label. And
why that's so, well, there we go. Why that's so important is, now I can hear myself 3 times. Why that's so important is because basically it's super fast moving science. So it's science that gets from the bench to the bedside pretty quickly. And stem cells set the stage, but other technologies, CRISPR probably being one of the fastest transitions. It's highly complex and interdisciplinary, so it's not something that most of us learned about in school. We didn't take AP stem cell or AP CRISPR. And then, of course, it also raises a whole set of questions, and you just heard about some of those that don't have exclusively scientific answers. Meaning those are questions not just about can we do this, but should we do this, and what are some of the larger societal implications of this, if we can make it some of these edits. CRISPR is one that I want to highlight, mostly because we released a report, and Anita Baracharya was already mentioned. She co-chaired that committee for the National Academies. We issued our report last year on the future, the political, regulatory, and scientific future of CRISPR. So these are edits to the human genome that are partly, in some applications, somatic, meaning they just affect that particular patient. They can also be to the germline, which means the edits that we're making are being passed on to future generations, and any potential error we're making is going to be passed on to future generations, with a limited understanding at this point about how to roll these things back, or if it's even possible to roll those things back. One of the things that we said in that report is that as we think about edits to the human germline, that we really need to have a broad societal debate about the potential implications, right? So think about just a few, and Chris and others can speak better to this, potential applications. In California, people are working on injecting pig embryos with human stem cells to create chimeric embryos to potentially grow organs for transplants. These are organs that wouldn't be rejected, that wouldn't require a whole bunch of specific drugs for the patient and so on. But they do combine human stem cells with a pig embryo. And that may have ethical concerns for some of us. That may have religious concerns or raise religious concerns for some of us, especially those religions who have certain issues with particular animals and so on and so forth. And those are just scratching the surface. Think about, at some point we may or we will have the ability to edit skin color and to change skin color. We know from decades of social science research that most of us, if we admit it or not, consider lighter skin color better, quote unquote. And we would never admit that, but if you actually do implicit bias tests with large populations, that's what comes out. So is there going to be pressure at some point on parents to edit the skin color of their future children? And what about those of us who decide not to do that? Are there going to be new forms of discrimination? All of these questions seem pretty far-fetched, but they're not very far-fetched once you're actually able to do these things. And somebody in a 7.2 billion people population in this world is going to try to do this. At which point those questions affect all of us because, again, some of these edits will be to the germline. So I just want to end by saying I think the conversations that we're having here is exactly the kind of conversation we need to have, and doing this at the 20th anniversary is even better. But it's kind of those broader debates that we called for. We devoted a whole chapter of that report just to that, and I'm glad that we, as Wisconsin, have played a leadership role in having those conversations.
Thanks, Dietram. So as Laura said at the beginning, I'm a scientist here upstairs on the 4th floor, actually down the hall from Jamie's lab, and it's a great place to be. We do a lot of work with human stem cell derived cultures, and a lot of them are embryonic stem cell lines that were first derived, as in the experiments that Professor Bhattacharya just mentioned. And there's such rich resources for me as an engineer, as well as all of my trainees, in essentially building up new types of tissues. So as Dietram just said, we can go in and rewrite portions of the genome. We try to fix mutations. We try to make tissues inside the lab such that we can test new gene therapies, new small molecule therapies, new biological therapies, any candidate therapy before it goes into the patient. So we can tailor these types of therapies in the dish. And so that's one way. It's been an established way. It's commercialized by a number of companies here in Madison. Another, I think, also quite exciting prospect is actually taking some of these cells and putting them into patients. So there's been a number of trials doing this in the eye, and I think that's just really the tip of the iceberg. So there's also blood cells that you could engineer to fight cancer. These are thought to be a really kind of off-the-shelf type of paradigm where you genetically engineer a stem cell, one of these embryonic stem cells, to produce cell therapies for all cancer patients, for instance. And so there's active research strategies there. Part of the, I think, reason why I'm excited about this type of discussion is understanding and thinking about the boundaries that we should have in the lab. And I think Wisconsin has historically been very responsible in thinking about these types of experiments carefully. And a great example of that was 20 years ago when they first did some of these original derivations. And I think we're at similar points right now in terms of how to edit these stem cell lines, of course, to try to generate new types of therapies for terrible diseases like cancer and blindness and muscular dystrophy, et cetera. And how do we make sure that there's a robust and well thought out boundary such that we don't go into some of the types of scenarios that Professor Schäufele just mentioned. That's really I think a tough question, and one that's thought of is being thought by not only scientists here in the building, and on campus, but also bioethicists, science communication professors, the general public. It's really been pretty fascinating to see how kind of the broad community has come in and chimed in on where those boundaries and how those boundaries should be constructed. So hopefully we can at least touch upon that question today, because it actually does quite, I think it will guide how my trainees think about working in this field, how I personally think about where we should go, and how to responsibly do science in this type of domain.
Chris, I want to pick up on what you said about how Wisconsin does have a really rich history of thoughtful about how we do these experiments. We do have a stem cell research oversight committee, which Chris and I are on, and the committee is made up of scientists, ethicists. We have some members of the public. We have a religious leader. We try very hard to make sure that we get as more opinions in the research besides just the scientists. But I will say that may not be the norm throughout the country. I think that Wisconsin, as Chris said, is a leader in that, and because of the history of having Jamie Thompson here, we did start a lot of that dialogue, but there are differences between states, there are differences between research institutions, and there are differences between countries. And so this makes the framework difficult to interpret, I think, for both scientists and the public. And I also think it adds to maybe a lack of transparency about how the science is done.
Countries, one of the things particularly telling, I think, for the effort around CRISPR, so in 20 acknowledged, and so it's in DC, it brought Academy, Royal Society, German Academy, French Academy, US Academies, acknowledging for the first time that this is really a global problem, right? That regulating this, and I mean, let alone in different states, but regulating this within the US is not going to help, because the beauty of CRISPR as a technology, of course, has been that it makes editing easier, safer, and cheaper, so that it can potentially be done much more easily by many more players. And that's in many ways not a bad thing, but it is a somewhat problematic thing if you think about regulation. So really the solution to this will have to be some harmonization of laws, and that's unlikely to happen. But I think it was really productive to see an early conversation with a global set of players at the table. The committee that issued the report represented all of these, and there was in 2019, I think it's either in Hong Kong or Beijing, another global summit that China is hosting. So I think just echoing the spirit of what you were saying, I think the other scientific communities.
I think on that note, there's just so much, I think, interesting scholarship to be done, thinking to be done there, but there's certain things that I think are not being as robustly discussed as they could be. For instance, access, what happens downstream of doing all of this research. If you make some of these therapies and they cost $500,000 to put into each patient, it is really being accessed by only a part of the potential general population. How do you manage that at a global scale? That's almost a impossible question to tackle right now, but it is something that's in the back of I think many people's minds, and there's many other examples like that. There's these, I guess, NBIC type of technologies where I think stem cells is one example of it. It's just in some ways the task could be thought of as very large, but there are kind of incremental steps here that are moving, I think, in a productive direction.
I'd like to come back to a little bit of a conversation about human embryos, and really why we're all here today is because the original derivation of these lines came from human embryos, and I think that really sparked people really thinking about how they felt about that. I think even, actually most scientists spent a lot of time thinking about how they felt about that. Because it was so new, it wasn't Dolly the sheep, we're talking about human embryos. And as I said, and as we've been discussing, scientists and ethicists came together to make guidelines, but those are guidelines or laws, and they may not make the public feel any better. And so I know that there are a lot of other issues that maybe you can speak to about an embryo itself.
Yeah, and I think that especially when it comes to public reactions to I think we all fall somewhere in a range, let's say, for religiosity, right? And so it's not just a simple religious concern, it's also a moral concern. So depending on what you believe, differ probably to some degree in our understanding of when human life starts and so on and so forth, and certainly the scientific community and some religious communities differ. And it represents, I think, really well a question that doesn't have a purely scientific answer, kind of what I outlined earlier, right? But it also represents some really interesting dilemmas. And so for example, on the CRISPR committee, when we talked to religious leaders, one of the things that they were talking about, and a really good friend of mine, actually, both he and his wife are carriers of Huntington's. And so they went through IVF, and they had the lots of spare embryos produced, and eventually had 2 healthy babies. And it's great. But you produce a lot of spare embryos in order to do that later on may be used for stem cell research. They may be frozen. They may lead to, George Bush favored snowflake babies or adoption of these embryos. There are lots of options that you can go through. And here's where the CRISPR example comes in, or we can actually figure out how to edit the genome of a sperm cell, for instance, the DNA, and we don't have to produce spare embryos because we can basically change that mutation so that their children won't have Huntington's and won't be carriers. But in order to get there, we need to do research with human embryos, right? So you can see how you're trading off creating spare embryos with IVF or doing research on human embryos to no longer have to do that in the future. But you have to do one or the other. And so I think those are the complexities where we can all see that there is no single best answer to those. There is only an answer that we as society hopefully come up with and move on. And I think one of the mistakes and again, I think Wisconsin is a real exception here, but I think a number of scientists have made as stem cell was rolled out, is that they really felt that there was and we call this the knowledge deficit model in the types of research that I do, that the public just doesn't understand it. They don't understand how important this is, how great this is for patients, and once they understand that, they'll buy into it. And what that completely ignores are these ethical, moral questions that even all of us in this room, who probably are fairly similar in certain views on science, would agree on. And so the problem is that we really can only come up with a solution that we as a society agree on. That's not going to be perfect, but it's going to be the best one. And I think the second thing for embryos in particular that was problematic, and I'm still at the stage in my life where I've spent more than half of my life in another country, meaning Germany. I'm a year away from being at 50-50. But you can even see for Germany where the outlook, it's a governing coalition right now. The biggest party is still the Christian Democratic Union, and it has the Christian in its name. The way they look at embryonic research is very, very different. But they also realize the political realities. Their best scientists are going to the US or Singapore or elsewhere to do the kind of work that they're not allowed to do. So in addition to, and Anita mentioned stem cell tourism earlier, there's also this outsourcing of research that sometimes leads to. So I just wanted to put that on the table as well, because I think it's an outcome that comes from different standards, regulatory or moral, that different countries hold their scientists to.
One other trade-off that comes to mind is the idea of preventing some type of animal testing. So I've heard of these arguments where if you could generate, really great tissue-based and culture-based human models, we would reduce the need to do as many primate experiments or animal experiments. So, that's some sort of moral calculation about us in relation to animals and animal rights. And so I think there's very interesting constructions of what type of rights scientists should have, what type of rights patients should have in this kind of new technological age. The right to have a genetically related child in the Huntington's case is something that could be quite strong for many individuals, but then others may say adoption is fine as well. So the diversity of moral views here is significant.
I think just to jump in because that was a big topic in the committee as well, the right to a biological child, which most of us haven't necessarily thought about what the trade-off is that we're making, and some ethicists have argued that what we're doing is we're basically exposing the whole population to an experiment, right? We're editing the human genome in order to cure genetically inherited diseases for people who want to have their own quote unquote biological children, and that's the trade-off. And again, I think there's some real, and I think we outlined those, some real cultural differences also, where in some cultures that's just much more important, having biological children, than in others, where adoption is much more of an accepted standard. And so there's lots of trade-offs, and I think that's one that I would have not necessarily seen without those broader debates.
I don't know if that's where the conversation's going.
We'd like to open it up to questions. Maybe there are things that we didn't make clear, or maybe you just have questions about what we were talking about, or if you have questions about something else. We're here to answer really any questions you have about human stem cells.
This question isn't really about the ethical landscape. I realize that's very complex too, but there was no mention of spinal cord injuries. How close are we to being able to use stem cells to repair that?
I will say I'm probably the most familiar. I would say close, not close. I'm not sure where we are. I will say that I think spinal cord injury is an area where there has been a lot of progress. And there are animal studies going on with human stem cells trying transplants into animals, not just rodents, but also into primates. As you can imagine, there's a big difference between fixing a rodent spinal cord injury and a primate spinal cord injury, just in terms of size and what you're asking the cells to do. I'll also say that it's still in the research phase. I think that most scientists who work on this would agree with that. I think there are moving fairly quickly towards clinical trials. But I also know that there are, if you were to Google stem cell therapy for spinal cord injury, you would probably get results that included many stem cell clinics that would potentially provide you with stem cell therapy for spinal cord injury. And many of these are not real stem cells and really are not backed up by science. So in the scientific realm, we're still in preclinical animal models, but they are promising.
I mean, one note on that is that trying to regulate kind of the legitimate stem cell trials versus perhaps the less legitimate ones is come into the spotlight quite a bit. The FDA has issued letters and warnings to some problematic clinics. But then there's also, I think, to connect it back to some of the ethical landscape that we're talking about, there is some movement of having a right to try, even some of these perhaps less legitimate type of stem cell treatments. So there has, in some circles, there would be a criticism saying that the FDA is too protective, and it's shutting the door to potentially some breakthrough treatments, and so let's let people have a greater freedom to try some of these perhaps more experimental therapies. Japan, actually, regulatory system has moved a little bit towards that end as well, to open up a bit more, I would say, less go into patients before having such a high standard of preclinical evidence. So, there is a pretty tricky boundary there, and spinal cord injury, I think, is one of those places where that's being worked out.
Could you speak to the more ethical end of things? What are the exact ethical objections to embryonic spinal or stem cell research, and specifically in Wisconsin? I know the legislature has taken action in regulating it, and I don't really understand what's going on there.
Yeah, and I don't think it's just a Wisconsin issue. I think it's a much longer debate that's been going on, and that ties into some other political debates surrounding abortion and others as well that are highly partisan or divide the country along pretty strong partisan lines. And one, of course, has to do with a lot of basic research on human embryos, meaning research that takes an embryo, for example, to viability, but then not beyond, right? So you're basically destroying, as some people would argue, human life, and you're experimenting on human life. And the example that I also mentioned from the Salk Institute in California, where you then mix together human stem cells with a pig embryo, for instance, and there have been questions in the bioethics community. So what would happen, for instance, if we grow a human brain in an animal, right? Does that animal now have consciousness? Does that animal now have the rights of some parts of human rights and so on? But I think the biggest one is really the religious argument about when life starts, and what the rights are of using human life for purposes therapeutic or otherwise, and to really improve medical treatments. I think that came the strongest illustration of that, and some of you will remember that when George Bush 43 really promoted the adoption of unused embryos, of spare embryos from IVF, and then other parents carried that baby to term, and he had a picture taken of himself surrounded by these parents with so-called snowflake babies. So embryos that could have been used in research, but now were young children with their parents. And I think that illustrates really the tension, right, that some people see between using human embryos for research and or not doing that. As I said earlier, especially for some of those new technologies, the ability for us to not create spare embryos depends on our ability to actually use them for basic research. So we need to invest in basic research on stem cell research in order to avoid kind of these side effects of IVF. And so we're really caught in a catch-22 unless we can do that kind of research in the academic community. We're not going to. So I think that's how I would kind of broadly, but I think there are lots of other dimensions.
So I will just add that we may not have made it very clear. We may have assumed more knowledge than we should have, that when the human embryonic stem cells are derived, or when they're isolated, it is from human embryos, and all of the human embryos that are currently being used to isolate human embryonic stem cells are all leftovers from IVF, and that these embryos are donated for research, and they go through the normal donation, donated in an ethical way. And so another issue that came up was the concern that people would go through IVF so that they could make embryos for research. And that is not happening, but that was one misconception. And I will also add that in this state, much of the legislation that is aimed at research is specific to fetal tissue research, which is separate from the human embryonic stem cell debate, in that human embryonic stem cells are derived from embryos which are within the first week after fertilization, whereas fetal tissue comes from farther along in development, and that tissue comes from fetuses that are usually obtained through elective abortions. So they are distinct, but often muddled together, or at least in this state.
I think these discussions are important and necessary, but given the broad spectrum of cultures and religious beliefs and everything else, do you ever see them converging on a consensus, or will we just debate forever?
I actually think that IVF is a weirdly good example for how we've come together around a debate that there were a lot of concerns, and if you look at some of the early media coverage when IVF first became a real option, and then the first baby was born, and the doomsday scenarios that people painted about how this child would develop, and children since then, and I think we've actually come around. I think Anita earlier spoke to the idea of overhyping certain things. I think that's the way we can mess it up, right? And I think we've, for stem cells, sometimes done that a little bit. We've promised too much. We've promised this is going to cure Parkinson's, this is going to make Superman walk again, and all these things, and so therefore people should make these moral trade-offs. And then the moment we don't deliver on the hype, then that really carries over into the next technology. So I think the only way we can, scientific community, well, there's many ways the scientific community could mess this up, but I think that to contribute unproductively to the debate would be to overhype it. I think that would be the one way of not coming to consensus eventually. I think we've seen for stem cell with people like Orrin Hatch and others out of Utah, senator out of Utah, coming around and basically supporting this and saying, well, I have value concerns, religious concerns, but I also see the value trade-offs in terms of really pushing the medical frontier. And I'm somewhat optimistic that especially for CRISPR, I mean, where and some of the CRISPR applications rather, where the therapies are just, once they will work out, are just phenomenal. I mean, for Tay-Sachs and debilitating diseases that we just don't have a realistic option for other than editing the human genome. I think we'll see much more dramatic impacts, and so I'm typically not the optimist on this one. I am.
I mean, one example, again, going to the CRISPR story, was that there was a moratorium in 2015 when they first thought through the application to modifying the germline in embryos. There was a pretty universal call to stop and halt any type of genome editing work on human embryos. That lasted for a year or so. And I think it's one example of where kind of the scientific community, many parts of it came together to say we're going to stop and think a bit more carefully about where we're going. The criticism of that is that was all internally driven, and they ended up going wherever they were going to go anyway. But I think there's an opportunity, and there was a recognition actually through some of these summits to say that that was a responsible step. There's more that we could do in consultation with the public, and that's kind of where we are right now.
Hi, it's my understanding that stem cells can be isolated or induced from living adults as well. And I'm curious how that source changes kind of these ethical questions.
So I think that's a really good question, and it's not something we brought up initially. After their initial isolation of human embryonic stem cells in 1998, about 10 years later, and within those 10 years, Jamie Thompson's lab and other labs around the world, including Shinya Yamanaka, spent a lot of time trying to figure out what is it that's so unique about a human stem cell, and can we make other cells look like human embryonic stem cells? And that occurred in about 2007, 2008 with what we called reprogrammed stem cells. And this is the ability to take a skin or blood cell from a living person, from an adult, a normal person, and reprogram those cells to look like human embryonic stem cells. And this was pretty amazing that you could really turn back the developmental clock. And yes, these cells are extremely valuable and are very much like human embryonic stem cells. They're not exactly the same. I would say they have some pros and cons. One of the advantages of these kinds of adult human stem cells is that they can be taken from people with different diseases and used. Then you have disease-specific stem cells, and you can use these to study how that disease might happen. On the other hand, they are not the same as human embryonic stem cells because they kind of remember where they came from. So they're not exactly the same as human embryonic stem cells. So for some studies where you want really blank slate stem cells, they wouldn't be so good. So for example, some of the therapies that we talk about, there's a lot of debate about, do you use these more adult reprogrammed cells that may carry baggage from where they came from? Or do you use the blank slate human embryonic stem cells that have more ethical concerns?
Could you elaborate a little bit on the issue of the stem cell clinics that you say use illegitimate stem cells? Just wondering where they derive their stem cells from.
Well, so there's many types of clinics like this. I guess probably one notable example that made the front page of the New York Times, for instance, was this clinic in Florida. I think it was called Sunshine or Sunrise Clinic, where they took essentially a bone marrow aspirate or a fat biopsy and injected it into the back of the eye to say that they would cure some type of vision disorder. And these patients ended up having pretty bad side effects from this type of treatment. And it turns out that this was kind of classified as a medical procedure, inpatient, moving cells around in your own body that does not require a IND from the FDA to go through the normal clinical trial type of regulatory path. And so there are ways where you can minimally process human tissue that comes from a routine medical procedure like liposuction or bone marrow aspirate or from just taking blood and put that in other places in the body. And so these can be advertised as stem cell treatments. If someone was to take a close look at what exactly was inside the syringe when they injected these cells, it's very likely that there may be no stem cells in there. Many of these types of clinics talk about adult stem cells. So this is quite distinct operationally from the embryonic stem cell derived therapies that have arguably now 20 years of development behind them. But that's probably one of the most extreme type of scenarios where essentially you're injecting fat cells into the eye and say that it's a stem cell therapy.
I will just add that I think that it's hard for people to know the difference between something that might be real and something that won't. And I think that's a lot has to do with using the term stem cell. I think when we talk about human embryonic stem cells, these are true stem cells. they are able to divide and grow indefinitely, and they can turn into all cell types of the body. There are many other stem cells that might be in different tissues, as Dr. Saha pointed out, and these are tissue-specific stem cells or adult stem cells that can't turn into everything. They can really only turn into the cell of the organ where they came from. And a really good example is bone marrow and bone marrow transplants. These kinds of stem cell transplants that are used for different kinds of leukemia are fantastic in that these are blood stem cells that are making more blood. And that strategy works. And we've been doing bone marrow transplants for a long time, even before human embryonic stem cells. So I think there is confusion about the term stem cell, and that the confusion that people may have is taken advantage of in some of these clinics.
So picking up on that conversation, it seems like it's theoretically really regulatable. Like there could be just a poster that says, this is possible, this isn't possible, claims to this effect aren't valid. So, I mean, scientists have some consensus about where the state of the art is. Is it a political barrier at the FDA level, or what's your understanding of why there is this possibility of confusion of therapies?
That. So I'm on this forum on regenerative medicine by the National Academies, and this topic has come up over the last 2 years. And there are these types of posters and guidelines, and there's even thoughts of media campaigns having commercials and having the doctors be educated. And so there's many ways, and I think there's really creative thinking on trying to address it that way. One of the major questions is why doesn't the FDA just stop some of this activity? And I think they're thinking about that. And one response that I don't think came from FDA, but they say there's so many of them that it would take a lot of their resources away from other activities to enforce this type of treatment. And if there was really true harm, like the example in Florida that I was talking about, there are other mechanisms that you could take legal action. That's kind of a reactionary approach. It might be an American approach. But I think there's a lot of ways that I think this could evolve, and I do think it will probably evolve pretty seriously over the next 5 years, because there are, when one looks at the U.S. and where these clinics are located, you can see hot spots. There's a lot in kind of Texas, Florida, California, and there's more, and it's been growing.
My question is a little bit wider than that, and it piggybacks on that. I think the genie's out of the bottle on this. It isn't just the United States. The entire world is working on this. I know of a company in Mexico that's doing private work, and 2/3 of their employees are bioethicists to look at the effects of what they're doing. What are the effects down the world to people who are having their lives changed by either having their lives extended or their diseases changed. So I'm feeling like we're a little bit, maybe the United States can regulate this, but the world is going on and doing their own things.
I think that it's a problem. You're right. We can only, it really comes back to what we've initially talked about at the beginning about having some framework for how to move the science forward. We also need a framework for how to be able to handle this explosion of stem cell clinics and be able to figure out which ones are legitimate and which ones aren't. And it is not easy to find a framework within the United States, much less the whole world.
And there's also, I'll jump in on one other thing, and that's the interpretation of what the Europeans would call the precautionary principle, right? And I think Europeans have long approached, and they have a certain appreciation for the nanny state that we don't have in the U.S. But so what they've done is they really put the burden of proof on corporations or on academics to show that something is safe before it can go to market, hence the precautionary principle. And in the US, we've often taken the opposite approach, meaning stuff is legal and we're able to do it until you can actually prove that it does harm. And the FDA that still takes clinical trials for a lot of these applications through the RAC, the Recombinant DNA Advisory Committee, which, as you can tell from the name, was created a long, long time ago, long before just this explosion of new research. And so it's in part also a bandwidth issue. And I mean, that's, the clinic example that Chris mentioned is just a really good one. We just don't have the infrastructures in place, partly because culturally we haven't done it, and partly because we haven't responded to some of those new challenges. I think that's part of the problem. So I do think there's a little bit of a unique U.S. difference from certainly some of our funding competitors in Europe that are spending equal amounts of money on that kind of research.
We can take 2 more questions.
At this stage, are there any, for want of a better term, diseases that have been found that stem cells just won't work? That is, efforts have been made and it looks like stem cells are simply not a useful approach to dealing with them.
So I think I don't necessarily have a specific disease, but I will say that when we think of stem cell therapy, usually we think of using cells derived from human stem cells to replace disease cells. So cells that have died because of a neurodegenerative disorder, for example, in Parkinson's, or cells that aren't working properly. So maybe islet cells in diabetes. But this kind of cell therapy, cell replacement, is not necessarily good for a disease like cancer, where the problem is that there are too many cells, and putting more in will not really help. Or since I work on the nervous system, I get questions about using stem cell therapy for autism or for other psychiatric disorders or Alzheimer's. And there, we don't have a good reason to think that putting in new cells will make a difference. And I think there's also the possibility in some diseases that there's something about the environment that the cells will go in that will make the new cells have the same disease. So I think there are a lot of diseases that stem cell therapy is not good for.
One addition to Anita's great answer was there's a story in the newspapers recently about cardiac stem cells and a retraction, and this is a high-profile example from Harvard where there was an original claim that the heart had stem cells that could be taken out and then used to regenerate the heart. It turns out that tissue doesn't really have stem cells in the adult, at least from the best studies that many other people have done, and they could not replicate that finding. So, it is also the case that certain tissues just don't regenerate very well in the adult, and if someone says you're going to have cardiac stem cells put into you, you should ask them many questions.
My question is, with the Right to Try Act approved, will it slow down the stem cell development in the US, and will it actually disrupt the ethical landscape that all the universities and FDA is trying to build around stem cell science?
Is the question if it would slow down development?
If it will slow down the stem cell development, being it's very expensive, so the scam artists will actually bring in therapies that don't work and try to sell it at lower price and the legitimate ones will not make the market?
Will it incentivize illegitimate science?
I think the common refrain that this is a more risky path, and they point to gene therapy from the early 90s as perhaps not doing some of their experimentations as carefully as they could have, and ending up having some, very high-profile adverse events that really slowed down investment in the field and slowed down the development of new gene therapies that some people say just started recovering in the last year or two. So, there's really a balance here of how much do you give patients the right to try perhaps cell therapies that are more risky, and how much is society broadly and also the capital markets and the investors willing to invest in a type of technology that may actually end up, unfortunately, killing some patients in the process. So these are very, very tricky questions, and going to, depend on each technology, each culture, each, I think, kind of threshold for tolerating these types of risks.
And just to add to that, I think is also, you're asking a very abstract question about what it will do to the system and to kind of broader perceptions. The problem that I think Chris vocalizes really well is you have to at some point explain that trade-off to a parent whose child has an illness and wants that treatment, and that parent might be you, right? And now society tells you cannot have this. And it was at that first summit that we mentioned earlier now a couple of times, a mother went to the microphone, and there are all these ethicists sitting on stage arguing, this may have these adverse effects for society. And she tells the story of her son who lived for about a week with seizures and couldn't obviously speak, being this young, but looked at her and she said, his eyes would say, "Mom, help me." And then she turns to the stage and is like, "If you have the technology, just frickin' do it." And the whole room is clapping and everybody gets the story because it's but it's directly at odds with some of the right for that parent to try is directly at odds with some of the downsides that you mentioned. But I think that's the difficult part of it, that every single one of us can very much imagine being in those shoes. And I think that's the difficult part of those conversations that we need to.
Thank you. I think that's a great way to end, and emphasizing the balance that we all bring to this field. So thank you very much, and we'll take extra questions if you have them. Thank you. Have a good evening.
Thank you.